So in this video, we are going to cover the oxidation fermentation, or what we call the OF test. And this is activity 5-2. And the first part of this will cover day 1, meaning the setup of this experiment. And then the second part of the video is going to cover day 2, looking at the readout for this experiment. And so let's talk about this experiment. So for the oxidation fermentation test, the purpose of this test is to determine whether bacteria oxidize and or ferment glucose. So we just looked at our phenyl red test, and in the phenyl red test, we were determining if bacteria could metabolize a specific carbohydrate. In the phenyl red test, we weren't differentiating based on oxygen requirements, meaning that bacteria could have oxidized the sugar, it could have fermented the sugar. All we were doing in the phenyl red broth test was determining if bacteria can metabolize the sugar. In this test, now we're getting more specific, and we're looking at, can they use glucose in the presence of oxygen, which is our oxidize, or do they use glucose in the absence of oxygen, which would be to ferment the glucose. And so now we're getting a little more specific in terms of the oxygen requirement. And so let's look at how this works. So there are 3 bacteria that we will use in this experiment. All 3 are gram-negative rod bacteria, just like we saw for the phenyl red broth test. So we have our Escherichia coli, our E. coli, again, found in the intestines. We have Pseudomonas aeruginosa, which we will abbreviate PA. Pseudomonas aeruginosa is the major cause of death in burn victims. So we have Pseudomonas aeruginosa, which we'll talk a lot more about coming up. We have Alkylogenes faecalis. So again, Alkylogenes faecalis. Faecalis refers to feces. Again, this is going to be a bacteria that's found in the intestines. And so these are the 3 bacteria we will test in this experiment. So the way that we would do this is that this would be done in groups of 4. And for the OF media is basically going to be what we call an agar deep. And what that means is that the broth is not fully a liquid, but it's also not a solid like a plate. It has a low concentration of agar. And that low concentration of agar is there for 2 purposes. 1, to help seal out oxygen so that we can have 1 tube that is anaerobic. And 2, the other purpose of that is that it allows us to determine motility. Basically meaning that it allows us to determine if bacteria are motile or can move. So the way that we would do this experiment, because this is an agar deep and because it's a semi-solid, we need to inoculate, not using a loop. A loop is used to inoculate with a broth, but for an agar deep, we need to inoculate with a needle. And to do that, we need to do a stab inoculation. So what your group would do is that your group would have 6 OF tubes. So 6 total. You would label each test tube with initials, lab day and time, so Monday, Wednesday AM, Monday, Wednesday PM, etc., and then the initials of the bacteria. So for each organism, like E. coli, for example, you would have 2 tubes that would be labeled with EC. You would have 2 tubes that you would inoculate with E. coli. You would have 2 tubes that you inoculate with Pseudomonas aeruginosa. You would have 2 tubes that you inoculate with Alkylogenes faecalis. And the reason for that, the reason that you need 2 tubes per organism is that 1 tube is going to get mineral oil. And the mineral oil is used to seal out oxygen, meaning to exclude oxygen from getting to where the bacteria is. This tube would be what we call our anaerobic tube in the absence of oxygen. That tube would be our fermentation tube. This is to determine if bacteria can ferment the sugar. The tube without the mineral oil would be our oxidation tube. It would be our aerobic tube, meaning oxygen can get in. And this tube will allow us to determine if bacteria can oxidized glucose. So that's why for each organism, you're going to need 2 tubes. One to test aerobic, one to test anaerobic, right? So you have to have 2 tubes per organism. So what you would need to do is you would use your aseptic technique and you would pick up bacteria with your needle and then you would stab inoculate. And when you stab inoculate with your needle, you go straight down and you go about 1 centimeter from the bottom and then go straight back up. And you're going to do that for 2 tubes per organism. So I would inoculate this one with E. coli. I would flame sterilize my needle. I would pick up more E. coli, and then I would stab inoculate this tube. So I would inoculate 2 tubes per organism. Now, for those 2 tubes, one of the 2 tubes, I'm going to add mineral oil to. And when I add mineral oil to the tube, I have to make sure that I don't contaminate the dropper. So I don't place the dropper inside the tube. I hover it over the tube and I add the mineral oil so that I don't contaminate the dropper and I make sure that the dropper stays free of contamination. So I have a video that is going to demonstrate how to set up our OF test. So in this experiment, we are going to do our OF test, which stands for our oxidation and fermentation test. And so what we're doing in this experiment is to determine if bacteria can oxidize and or ferment glucose. And what that means is that for oxidation, this is going to happen in the presence of oxygen. So we'll have 1 tube that is aerobic and will be exposed to oxygen. And we will have 1 tube that we will make anaerobic, meaning without oxygen. And the way that we're going to make this tube anaerobic is after we inoculate the bacteria, we're going to put a layer of mineral oil across the top, and that's going to seal out the oxygen so that that tube is anaerobic. So what we need to do is we need to take our organism and we need to inoculate it into 2 tubes, because 1 tube is going to be aerobic, 1 tube is going to be anaerobic. And in this experiment, we actually will be doing this with 3 organisms, but for the sake of demonstration, I'm just going to demonstrate with simply 1 tube, 1 organism. So I'm going to do this with E. coli. So what I'm going to do is that this is a agar deep. Notice that it's not a broth and it's also not a liquid. It's in between. It has a low concentration of agar in it. So when I go to inoculate a deep, meaning it's not solid, but it's not a broth, I need to inoculate that with a needle. So my needle is going to be the tool that I'm going to inoculate this with, because I need to do what's called a stab inoculation. So I can't use a loop, because a loop would be used for if this was a broth, but we need to stab inoculate. So I'm going to take my organism, I'm going to loosen the cap. I have my flame going, so I need to flame sterilize my needle, starting with the base, working towards the tip. And I'm going to let that cool. Now, I'm going to pick up my organism, and I would make sure that my tubes are labeled. They would be labeled with my initials, the initials for the organism, so in this case, EC for E. coli, and then the date, 9/4/2020. So I'm going to let my needle cool, and then I'm going to take my loop hand, palm up, hold the cap between my pinky and ring finger, flame. I'm going to go in with my needle, and I'm just going to pick up a little bit of bacteria. I don't need a lot. Flame sterilize, put the cap back on. I need to inoculate my OF broth. So hold the cap between your pinky and ring finger, flame it, and I need to stab inoculate. I go about 1 centimeter from the bottom and then come straight back up, flame, cap, and then flame sterilize my needle. So notice that I moved it within the rack because I'm done with that, but I still need to inoculate 1 more. So I'm gonna, while my needle is cooling, I'm going to pick up more bacteria to inoculate the second tube. So my needle's cool, tap between my pinky and ring finger, flame it, go in, pick up some of the bacteria that's on the slant, flame it. Then I need to inoculate my other tube, flame it, and I'm going to stab inoculate, straight down and straight back up, about 1 centimeter from the bottom, flame. And then I'm done with my needle. And I can set that down. So now I've inoculated my organism into my 2 tubes. It's the same organism in both tubes. I need one to be aerobic. The aerobic one is going to be the one that has oxygen. That is going to be the one without mineral oil, meaning this tube I'm just going to leave as is. This will be my aerobic tube. To the second tube, I need to make this anaerobic. And so again, to do that, I'm going to add my mineral oil, and I just need a good layer across the top to seal out any oxygen. So I'm going to loosen my cap on my mineral oil. I'm going to take my tube, and it's really important when I add my mineral oil that I hover the dropper over the tube. I don't want to stick the dropper in the tube because I don't want to risk contaminating my mineral oil. So watch how I do this. So I take the cap off, flame the opening, and I'm just going to gently add enough oil to make sure that there's a good layer, flame, cap back on. And so you'll notice that I have a nice little layer there, and that layer of mineral oil is going to be there to seal out the oxygen. And so this tube is now my anaerobic. This tube is going to test for fermentation, and my tube without the mineral oil is going to test for oxidation. And so I will do that for all of my 3 organisms. I will inoculate in pairs. So for each organism, I will inoculate into 2 tubes. And then for one of the 2 tubes, I'm going to add the mineral oil. That's testing my fermentation. And I have my 1 tube without my mineral oil. That's testing for oxidation. And so this is the setup for my OF test. So now we're going to talk about the readout for our OF test. And this would be done 48 hours after we inoculate the agar deeps. So in order for us to understand how the readout works for this test, we need to first understand what is in the media. So the recipe for the OF test is going to be found on page 296 of your lab book. And the name of the media is basically OF media. That's it. That's a simple name. It's just called OF media. And if we look at the ingredients that are in the media, the first ingredient that we have is going to be our pancreatic digestive casein. Now, you might recall that we saw that same ingredient in our fennel red broth test. And remember that we said that casein is a protein. And if we have a pancreatic digestive casein, it's that protein mixed with pancreatic enzymes. The enzymes are going to digest the casein. They're going to digest the protein. And we would refer to that as peptones. Peptones are partially digested proteins. This ingredient is in the media because it is food for general growth. So it's food for general growth, and as a result, this pancreatic digestive casein would make this media complex or undefined media. Because again, peptones can vary from batch to batch. So we have our peptones, then we have our sodium chloride, and sodium chloride again is there for salt balance. Again, it's to regulate so that too much water doesn't go into the bacteria and too much water does not go out. So it helps to regulate water movement within the cells. Next, we have our dipotassium phosphate. Our dipotassium phosphate acts as what we call a buffer. And the purpose of the buffer is to help keep the pH constant because cells don't tolerate big swings in pHs. And so we add buffer to the media to help keep the pH constant. Next, we have our agar. And our agar is a thickening agent. And the agar is there for 2 purposes. Again, one, to determine motility, to see if bacteria can move away from the stab line. And 2, the agar is there to help exclude oxygen. basically meaning to help seal out oxygen so that we can have a tube that is anaerobic. Now, in our agar deep, there is a low concentration of the agar. So again, it's not as solid as a plate or a slant, but it's also not so low that it's a broth. It has a low concentration, and as a result, the media is a semi-solid. So it's semi-solid, which is why we inoculate with a needle and we do our stab inoculation. The next ingredient we have is bromothymol blue. Bromothymol blue is a pH indicator. Again, it's going to change color depending on the pH of the solution. So if you remember in the video watching the tubes started out green. What does that likely tell you the pH is like for the green? Well, most bacteria are neutrophiles. They like to grow at a neutral pH. So green is going to be neutral. You might also recall from the fennel red broth video that for our pH indicators, I said that there is one color that usually indicates acidic, and that is yellow. So in our bromothymol blue, yellow is acidic. Again, for all of our pH indicators except methyl red, yellow is acidic. So yellow is going to be our acidic color. Green is our neutral. That is our starting pH of our tube. And blue is going to be alkaline. So one of the things that you'll notice is the neutral is the color in between. So like if you've ever painted before, right? If you take blue and yellow and mix them together, the color that you're going to get is going to be green. In our fennel red, right? Yellow is acidic, pink is alkaline. Mix them together, you get orange, which is your neutral. So your neutral is going to be the color in between. So in the case of bromothymol blue, yellow is acidic, blue is alkaline. That's why it's called bromothymol blue. The other thing you can think about is that usually the name, so the fact that it says bromothymol blue, that blue is going to be the alkaline color. Fennel red, red is the alkaline color. When we get to bromocresol purple, purple is the alkaline color. So it makes it a lot easier just by looking at the names. That will help tell you what those colors mean. So whatever you see in the name, that's going to be the alkaline color. And again, yellow is going to be acidic. The only exception to that is going to be when we look at methyl red. Other than that, everything else will follow that rule. So we have to have our bromothymol blue to detect changes in pH. And then the last ingredient that we have in the media is going to be glucose. And glucose is going to be our substrate for oxidation or fermentation. So it is what we are testing for the oxidation or fermentation of. So we have to have the substrate in the media so that we can determine if bacteria can oxidize and or ferment glucose. So we have to have glucose in the media. So if we look at our OF test, so we're starting with glucose. Glucose is our substrate. Glucose is a monosaccharide. It's one sugar. Now, when we talk about cells, bacteria can use glucose aerobically. And when it uses it aerobically, it's going to oxidize it. So oxidation, think oxygen. That means that the oxygen is present. So some bacteria can use glucose aerobically. And our enzyme in this case is going to be various endoenzymes. So again, in this test, we're not testing one specific enzyme. We are just determining if bacteria can oxidize and or ferment glucose. There's a variety of enzymes that are involved in this procedure. So again, we're not testing one enzyme. We are just testing if bacteria can do this process, which involves a variety of enzymes. So my substrate is my glucose. My enzymes I'm going to record as various endoenzymes. Now, aerobically, again, aerobic is referring to oxygen. that means that the bacteria is going to oxidize the glucose. In the case of fermentation, fermentation is without oxygen. So without O2, bacteria would do fermentation. That is an anaerobic process, right? Anaerobic means without oxygen. So bacteria have choices. Some bacteria might only oxidize. Some bacteria might only ferment the sugar. Some bacteria can do both. Some bacteria can do neither process. They don't use sugar at all. That's what we're testing in this experiment, is we want to see, do bacteria oxidize and or ferment the sugar? Now, the oxidation tube is going to be the one with no mineral oil. The one without mineral oil is basically going to be able to get oxygen and therefore is our oxidation tube. Our fermentation tube is the one with the mineral oil. So the one with the mineral oil, the purpose of the mineral oil is to seal out the oxygen, to prevent the oxygen from getting to where the bacteria is growing. That will be our anaerobic tube. Now, in terms of our products, so when bacteria use glucose aerobically, they will produce a variety of acids. So pyruvic acid, we'll look at a lot of other acids that get produced during the citric acid cycle, which is also known as the Krebs cycle. Lots of acids can get produced during aerobic respiration. In the case of anaerobic or fermentation, fermentation will produce acids, alcohol, and plus or minus gases. So some bacteria will produce carbon dioxide gas, others will not. So on your sheet, your products, you're going to have two sets of products. So for fermentation, your product is going to be acids, alcohol, and plus or minus gases. For your oxidation, your product is going to be simply acids. And so this is our test. We want to determine if bacteria can oxidize and or ferment glucose. So let's go over our results. Let's talk about what our results would look like. So remember that when we do this, we had 3 organisms that we were testing. We have Pseudomonas aeruginosa, we have E. coli, and we have Alkaligenes faecalis. So one set of results would look like this. So notice the tube without mineral oil is yellow on the top, green on the bottom. The tube with mineral oil is green throughout. So remember, what do those colors tell us? And the answer is that yellow tells us acidic, right? Meaning that the pH is low. Green means that it's neutral. Remember that these tubes started out green. So in this tube, which is our aerobic tube, notice we have yellow at the top, which tells us that this is positive for oxidation. So in this case, our bacteria was able to oxidize the sugar. On the flip side, bacteria were not able to ferment the sugar because notice that the pH stayed neutral. And in fact, the bacteria didn't grow without oxygen. So in this case, our bacteria is going to be our Pseudomonas aeruginosa, the one that we label PA. And our interpretation would be that this is positive for oxidation, because again, it's yellow at the top. That's the aerobic. It did not turn yellow at the bottom because that becomes anaerobic. So positive for oxidation, but negative for fermentation. So this organism can oxidize the sugar, but cannot ferment it. We would say that this bacteria is a strict or obligate oxidizer of glucose. So it is a strict oxidizer of glucose. Now, if we look at the next one, so notice that the next organism, it's yellow throughout for the oxidation tube, and it's yellow throughout for the fermentation tube. So what that tells us for this is going to be our E. coli, so Escherichia coli. And did bacteria oxidize the sugar? Yes, because it's yellow in the tube without mineral oil. Did bacteria ferment the sugar? Answer is yes, because it's yellow throughout the fermentation tube as well. So this one is positive for oxidation, positive for fermentation. So it's able to both oxidize and ferment the glucose. And so we would say that this bacteria is an oxidizer and a fermenter of glucose. What we call this type of organism is that we call it a facultative anaerobe. And a facultative anaerobe, what that tells us is that bacteria grow with or without O2. So when O2 is present, they will grow. When O2 is absent, they will grow. So they can grow under either condition. However, they will grow better if O2 is available. So for these organisms like E. coli that can do both, they will grow better when O2 is available. And the reason for that is that through oxidation, bacteria can get about 30 to 32 ATP per glucose. When bacteria do fermentation, they're only going to get 2 ATP per glucose. So for organisms that can do both, they can grow in both conditions, but they will grow better when O2 is available because they get a greater yield of ATP. So again, we call this a facultative anaerobe, meaning that it can grow anaerobically if it has to, meaning if O2 is not available. The next example that we have our aerobic tube, which is blue on the top. So we write this B/G. It's blue on the top and it's green on the bottom. The fermentation tube, the anaerobic tube, is green throughout. This is going to be the result for our alkylogenese faecalis. Alkylogenese faecalis is going to give us this result. Did alkylogenese faecalis oxidize the sugar? Answer is no, it's not yellow. Did alkylogenese faecalis ferment the sugar? No, it's not yellow. So it's negative for oxidation, it's negative for fermentation. So we say that this is a non-oxidizer and non-fermenter of glucose, right? The bacteria did not use the glucose either aerobically or anaerobically. It didn't use it at all. And remember back to our phenyl red test, I gave you a term for an organism like that. And that is that organism would be referred to as non-saccharolytic. Non-saccharolytic. Saccharide again means sugar. These are organisms that don't use sugar. They don't use sugar, but one of the things you can tell is that in this case, because the top is blue, what did bacteria use as a food source that raised the pH? And the answer is that they use proteins. They utilize the peptones, but only aerobically. And so when grown aerobically, they will utilize the peptones and produce alkaline products, which turns it blue. So this would be our result for alkylogenese faecalis. Again, remember that alkylogenese faecalis evolved alongside other bacteria in the gut. And as a result, they evolved to use proteins as a food source so that they're not competing with organisms like E. coli, which are going to use the sugar. So this is just an example of evolution in action. Bacteria have evolved to have mechanisms to be able to partition and to divide up resources so that they're not all competing for some common resource. They can use different food sources so that they both can survive in the gut. And so this would be our result of our OF test. So again, what you're looking for in this test is yellow. If you see yellow, that means that the pH is acidic, and the pH becomes acidic when the sugar is being used. So in the case of Pseudomonas, it's only yellow in the aerobic tube. So this is positive for only oxidation and negative for fermentation. Again, it's a strict oxidizer of glucose. For E. coli, it's yellow throughout in both tubes. positive for oxidation, positive for fermentation. Bacteria can both oxidize and or ferment the glucose. In the case of Alkaligenes faecalis, none of the tubes are yellow, which tells us the organism is non-saccharolytic, it doesn't use sugar. So this is just showing you these results closer up so that you can see what this looks like, so you can see the colors more apparently. And so again, notice for E. coli, both tubes are yellow. It's able to oxidize and or ferment. Pseudomonas aeruginosa, it oxidizes but does not ferment. Alkaligenes faecalis does not oxidize or ferment. It does neither, but it does use proteins aerobically. So notice it turned blue on the top of the tube where O2 is available. And so this would be your result for your OF test. Now again, you don't have to memorize the results for each organism, but if you were given tubes, you should be able to interpret those tubes. You should be able to tell me what it means if you see both tubes turning yellow. Or what does it mean if the aerobic tube turns yellow and the anaerobic does not, etc. So this would be your OF test and how you would analyze those results.